figured out the technical problems. We're good today. All right. So we have a quiz on Friday on viruses. We're going to finish viruses today. All right. You'll see there's probably 5 added slides in today's presentation that I will post right after today's presentation, along with the video for today's presentation. I posted Monday's video, so it's up there. I did that this morning, so we'll have. Hi, Nick. Okay, so you'll have everything set up. All right? Questions? Quiz on Friday on viruses. All right, so we're going to finish. We went over the lytic and the lysogenic cycle of bacteriophage, right? And the lytic and lysogenic cycles are good examples of how viruses work, okay? Whether it be bacteriophage or even animal viruses, all right? They have some similarities. I'm going to see that in a second, all right? Parts of animal viruses, and you'll see that the difference between bacteriophage and animal viruses is how they get into the cell. All right? Why? What's the difference between a bacterial cell and a animal cell? What? A cell wall. All right, which is present in prokaryotes. All right? There's not a cell wall present in animal viruses. In animal cells. In animal cells, they have a more selective cell membrane. Okay? So the mode of interaction changes, right? So there has to be some interaction that occurs at the level of the cell membrane instead of at level of the cell wall or the outer membrane of a bacterial cell. All right? So before we get into that, we're going to go through the classification of viruses. And these next 2 slides, you're expected to know the different types of genomes of viruses and examples of each. All right, so I put this up in a second. What possibilities do we have for genomes for viruses?
DNA or RNA.
DNA or RNA, nucleic acids. All right, that's what genomic information is. All right, and then you can break down animal viruses and whether or not they are enveloped or not. What is an envelope?
Covering for the.
So it's an outer covering of beyond the capsid proteins. Okay? So whether or not viruses have an envelope distinguishes them. Right? So if you were to draw up a dichotomous key, you could say RNA, DNA, right? All those viruses, and then whether or not they're single-stranded or double-stranded RNA, DNA viruses, and then whether or not they have an envelope or not. All right, so you can kind of follow it down that way. Okay? Here's classes of animal viruses. All right, and basically what you see is whether or not they're double-stranded or single-stranded DNA. All right, double-stranded DNA, adenovirus, herpes virus, all right, and papoviruses. Okay, do they have an envelope? Some do, some don't. All right, pox viruses do. Herpes viruses do. Right, adenovirus does not. And then to add to that, let's look at the diseases that they cause. Because viruses are pathogens. Right, they cause human disease. All right, so adenoviruses cause respiratory viruses, and they also have been associated with tumors. Right, adenovirus is one of those viruses that molecular biologists have utilized and changed their genome so they're no longer infective to humans. All right? But they still infect cells. What's the advantage of that? How can you use that in the lab, per se? Or in therapy, per se? What happens to viruses? When I expose cells to a virus, what happens? The virus gets in, right? What gets into the cell? The genome. So if I had, say, a patient who has cystic fibrosis, a respiratory disorder, where their gene, there's a one particular gene that's disrupted, but if I put the normal gene inside of that adenovirus, right, and then I expose their lung cells to that adenovirus, though that RNA or that genome, the DNA genome gets integrated to the cells, now what's going to happen to those cells? Ideally they would produce the correct protein, which would be a way of treating cystic fibrosis. Okay? Seems like a grand idea, right? A little simplistic in the way I put it. And there's a lot of obstacles that doctors and molecular biologists have to overcome in that sort of sequence. But that is the goal, all right? And they're looking at viruses as being a way of what we call gene therapy, all right? A way of using genes as a therapeutic, right? And delivering genes to cells that have abnormal gene function. All right, so we have herpes viruses. They cause herpes simplex, cold sores, genital sores, shingles, the chicken pox, all right, Epstein-Barr virus, which causes mononucleosis, and Burkitt's lymphoma. All right, Epstein-Barr virus, how many of you had mono? Okay, you have the Epstein-Barr virus somewhere inside your cells. It gets integrated and it holds on there. All right, it's somewhere inside the DNA at some point. All right, and then you have smallpox and cowpox viruses, which are also enveloped. All right, single-stranded DNA viruses. All right, parvovirus. How many of you have dogs? How many of you know that they are vaccinated against parvo every year, or as puppies? Yes. Parvo is a virus that causes intestinal discomfort and, for lack of a better word, explosive diarrhea in dogs, which can be fatal. All right, it's treatable. All right, you alleviate the symptoms and let the dog sort of get better on its own. Okay, but it can be fatal due to dehydration and intestinal bleeding. All right. Then we get into RNA viruses, double-stranded RNA viruses, reovirus, all right, such as a rotavirus or Colorado tick fever virus. All right, so they're viruses that cause human disease. Questions on this? Viruses. All right. The other families. So now we have single-stranded RNA viruses. Now the difference being here, what has happened up until now? We've had DNA viruses and double-stranded RNA viruses, right? So what's the typical progression in gene expression?
Well, those might get broken up by lysosomes because they're double-stranded, whereas single-stranded RNA might get through easier.
All right, so that's the sort of the defense mechanism from the cell, and you're correct. But what's the normal progression of gene expression? DNA to what? mRNA to protein. So in order for, say, a herpes virus to begin transcription of its genes and make more viruses, that DNA has to get into the nucleus, right? Be transcribed, form mRNA, and then eventually becomes, viral genomes and capsid proteins, etc. Okay? And that's the same, but when we have an RNA genome, there's a different mode that can happen. Okay? That RNA can be integrated into the cell, a single-stranded RNA, and automatically and right away be transcribed. Okay? So here you see a single-stranded RNA which serves as an mRNA, right? Versus a single-stranded mRNA which the genome serves as a template for our mRNA synthesis. All right, so there's two different modes of action, all right, or even a third. which we talk about a template for DNA synthesis. All right, so it sort of separates how that single-stranded mRNA or that single-stranded RNA is used. All right, so we have coronaviruses, togaviruses, flaviviruses, and they cause a whole host of different diseases. Common cold, SARS, you're familiar with SARS, right? West Nile virus, yes, hepatitis C, rubella. How many of you have, all of you have had measles, mumps, rubella shots, right? You are required to have them to get in school. Even if your parents are one of those people that don't believe in vaccination, you have to have it before you come here, all right? Measles, mumps, rubella, all right? And Eastern equine encephalitis, whether it's Western or Eastern, is passed by what? Equine encephalitis, I'm sure you've heard this in the news. What does it pass by?
Contact?
Not contact. Triple E.
Oh, mosquitoes.
All right, mosquito. So Eastern equine encephalitis. I know Triple E is probably the more well-known, but that's what it stands for. All right, and then there's Western equine encephalitis, and there's also another virus called St. Louis equine encephalitis virus. All right, so those are all passed by mosquitoes. Different modes of action. All of those diseases caused by viruses, and they all fit in that toga virus structure. Okay. Those viruses that use template for our mRNA synthesis include filoviruses, which is Ebola. You've heard about Ebola, right? Influenza, right, is an orthomyxovirus. I always end up with that one, right? Measles and mumps are paramyxoviruses, right, and they are enveloped. And then you have the rhabdoviruses, which is a rabies virus. Okay? And then finally, you end up with the last mode of action, which is a retrovirus, whereby the single-stranded RNA serves as a template for DNA synthesis. All right, so it goes backwards than the normal progression from DNA to RNA. It's going from RNA to DNA. All right? So what's the general term, or what's the general sentence you could use to describe viruses in terms of their genomic action or their action in general? They're pretty diverse, right? In how they get into the cell and or how they end up hijacking the cell in one way or the other, right? They're diverse in their genome. They're diverse in their action. So we're going to talk about viral envelopes, all right? An animal virus with an envelope uses it to enter the cell. Okay, if it does not have an envelope, there's a specific interaction between one of the capsid proteins on the virus and a receptor, okay, on the cell membrane. Okay? So that's how a virus enters the cell. There is a specific interaction between the virus and its target cell. All right? The envelope itself is a derivative of the plasma membrane of a previously infected cell. Okay? So what do you think it has in terms of structure? If it's a derivative of the plasma membrane, what does it look like? A lipid bilayer. Probably has what else? Some proteins in it. Okay? So that's exactly so it's going to structure itself like a cell. It's a way of masking the virus from outside immune function. All right? So if an immune cell is going around looking at cells and trying to detect whether or not they are foreign, it comes in contact with this virus, which is protected by an envelope. What happens? Is it going to be detected by the immune cell? No. It's almost like you having a fake ID. Okay? You're going out with all your friends that are 21 plus, right? You want to go too. You run into the guy at the door, and you have the fake ID. You gain entry because you have a fake ID. You have that identification that says you are 21 too. What, Ben? Don't have a guilty look on your face. Okay? But there are some special proteins, not just any portion of the plasma membrane that becomes part of the envelope. It is a selective portion of the plasma membrane. All right? So here's sort of an envelope virus's cycle. All right, so it gains entry into the host cell, the viral genome replicates, right? Depending upon the viral genome, right, it may serve as an mRNA, it may be replicated, it may be DNA, whatever it might be. Okay? However it ends up, what you see here is that the proteins are made, right, capsid proteins, but also envelope proteins are made in the ER. Right? So part of the genome has specific proteins that get embedded into the membrane that will determine that portion of the membrane becomes the envelope protein. Okay? Will become the eventual envelope to the cell. All right? Rest of the virus, capsid, and genome gets packaged. There's a collection of those envelope proteins in a certain area, and then the virus is sort of expelled by sort of an exocytosis that occurs. All right? So it's another way of hijacking the normal cellular process of producing proteins and expelling them out of the cell, and then producing which ends up producing an envelope virus. Okay? Questions on envelopes. All right? But the envelope itself is sort of determined by these glycoproteins that are from the virus. Okay? So RNA is a viral genetic material, broadest variety of RNA genomes. We have retroviruses, which are specific, and they use a specific copy of a protein, which is called a reverse transcriptase. All right? Which is coded for in their genome. It's not part of our genomes, right? We don't produce a reverse transcriptase protein in our cells. So that is part of the viral genome. All right? So then that protein is made in your cells when it's infected, and then it makes a copy of the RNA in the form of DNA. All right? Example, HIV. All right? And HIV is the retroviruses that cause the disease AIDS, acquired immune deficiency syndrome. All right? How does this work? All right? It's somewhat similar to the lysogenic cycle. All right? So HIV and retroviruses in general. Okay? What's the point of producing a copy of the RNA into DNA? Why not just infect cells?
To put it into the cell's genome.
So it gets integrated into the cellular genome. And that's why it usually takes a very long period of time to end up with infections with disease states over time. So what occurs is that copied DNA, like that viral genome that's copied into DNA, gets integrated into the host cell genome. What does that sound familiar to you? What does that sound like? Where a piece of DNA gets integrated, a piece of viral genome gets integrated into the host cell genome. Not a plasmid. Sounds like a bacteriophage in the lysogenic cycle. Right? That piece of that copied viral genome in the form of DNA gets integrated into the genome and now gets replicated as cells reproduce. Now one cell that is infected, 2 cells, 4 cells, etc., etc., etc. That genome gets passed on. Okay? So, and what do we call that in bacteriophage? That piece of DNA that gets integrated? It's called a prophage, right? In animal viruses, we call that a provirus. Sounds pretty familiar, right? Okay? Unlike prophage, though, a provirus is a permanent resident of this host cell. It now becomes part of the host cell genome. Okay, it doesn't go away. Then from that point on, the host RNA polymerase transcribes the proviral DNA into RNA molecules, making the genome and mRNA together to make the proteins and also package the genome, which then allows new viruses to be produced and then released from the host cell. Yes?
So is that, like last slide, kind of like a description of the retrovirus?
This is all retrovirus.
Okay.
Okay? So here's your this is, and we're specifically talking about HIV. Okay? So here's HIV. HIV is unique in that it's not just the genome that gets passed on from one cell to the another. It actually brings a copy of the reverse transcriptase with it. Okay? So what you see here is, here's your full mature virus, glycoproteins, it's enveloped. On the inside you have the capsid. The little red squiggles are the genome. The green dot is a protein for the reverse transcriptase. Okay? Enters the host cell, it gets uncoated. The viral RNA, immediately, first thing that happens is that RNA is reverse transcribed into a DNA RNA hybrid. Okay? So now it's a DNA RNA hybrid, protects itself, that then the DNA is replicated and replaces that RNA. RNA leaves, is removed, and now you have a double-stranded RNA which then gets integrated into the host cell genome, becoming your provirus. Over years and years and years, that provirus gets replicated along with other cells, and then the cell will also use our host cell RNA polymerase, make more viruses, right? RNA, glycoproteins, etc., and the reverse transcriptase packages them into an envelope and then sends it off as a new virus to infect even more cells. Okay? What you see on the right here is a scanning electron micrograph of HIV infecting a cell. All right, so here's your membrane, right? And then of a white blood cell, it then it gets phagocytosed and gains entry, right? And then that's this part. They don't show the rest. And then this is the budding of a new virus. You see this collection of sort of that dark area right about here is a collection of glycoproteins which denote where the new envelope is going to be. All right? And then you have new viruses being budded off. Okay? So now, one infected white blood cell, as long as it's living, will continue to produce more virus, will continue to divide, and produce more cells that have the provirus in it. Okay? Here's another graphical representation of this. All right, so you have integration. All right, so this is sort of how it works. And here's your RNA here, your mature entry, removes the envelope, then uncoated, ends up your RNA with the reverse transcriptase, DNA RNA hybrid, double-stranded DNA, integration, then transcription, translation, you end up with capsid proteins, envelope proteins, the reverse transcriptase, which all gets packaged into a new virus, a complete mature virus, which then can be enveloped and set off, right, assembled, and then released into the nearby to infect more cells. The structure of the retroviral genome. Okay? So three important proteins you see up there: GAG, POL, and envelope. Okay? Those are three genes that are important for glycosaminoglycans, GAG. Okay? GAG encodes the capsid protein. Envelope encodes proteins that will denote where the envelope goes. And POL encodes both the reverse transcriptase and an enzyme called integrase, which will allow that double-stranded piece of DNA to get into the host cell genome. Okay? Then there's a bunch of other things in red, what you see up there. They help modify host cell processes. Right? This in blue is your RNA genome of the retrovirus. Okay? Look at that. All right? Look at the RNA genome. And then tell me, think about it, talk to your buddy, your neighbor. What about that RNA genome is important? All right? Then you can answer this question. If you want me to put the genome back up, let me know. What? No, I can't. I don't have that capability to put them side by side right now. Here, I'll put up the genome back up for a minute. Okay? Look at it.
Are we looking at the blue thing?
The blue thing is the structure of the RNA genome. Okay, and then what you see in green is the parts of the genome that are the areas for those genes: GAG, POL, and envelope. Then you have the other parts in red, which are alternatively spliced, which allow for modification of host cell processes. Okay, so you're really looking at everything, but what part of the genome do you think is important? Okay, 10 seconds. Okay, so what feature of the HIV genome allows it to be replicated in the cell? D, the 5' cap. Why?
So it doesn't get destroyed.
So it doesn't get destroyed. What does it look like? It looks like a host cell RNA, right? What's the structure of a host cell RNA if I was to draw it out here? So I'll draw it in red up here, or black. Here's a host cell RNA. So that's the coding region. What else does it have?
5' cap.
Right.
Poly A tail.
5' cap and a poly A tail. Okay? This is 5', this is 3'. Right? Here you have the cap protein. Terminal repeats probably function as this end functions probably has a couple A's in it, so it looks like a poly A tail. So then what's that mean? So that genome enters the cell, it's not going to get degraded right away. Why? Because it looks like a host cell RNA. All right? It's a way for the virus to evade being destroyed. Okay? Okay, here's entry into now how the virus enters the cell. All right, up top, you see a scanning electron micrograph of RNA, HIV entering a blood cell, all right, or being budded off, and how the fusion of the virus occurs. All right, the way this occurs in cells in HIV is a distinct receptor that the virus interacts with, all right, called the CD4 attachment. All right, a CD4, it's a glycoprotein on white blood cells, all right, which is distinctive for human white blood cells. So you have this CD4, right? Here's your HIV membrane, and then you end up with this fusion protein. There's an interaction between these two, all right? When that occurs, this CD4 moves over to the chemokine receptor, and then it initiates an interaction whereby the HIV gets integrated into the cell. Okay? So it is a distinct, specific interaction with a protein on the cell surface, on the cell membrane, that allows for integration or entry of the virus into the cell. Okay? Some individuals actually are immune to HIV infection. All right? Because they have a disruption, a genetic disruption in that CD4 protein. So the interaction between HIV and the CD4 can't occur. All right? This is another area of therapy. All right? Another way of gene therapy. If we can change or disrupt the way or block the way HIV interacts with the CD4, we can prevent HIV infection. All right? By preventing its progression. Okay? Questions on that? All right? Now, viruses evolve. All right? They don't fit sort of the definition of living things because they don't reproduce on their own, but they do fit portions of the definition in that they evolve. All right? The reason, how do we know viruses evolve?
Because they're resistant to antibiotics.
Well,
over time.
we can't use antibiotics against viruses. Okay? Well, how do we know viruses evolve? How many? There's different strains. Okay? Different strains. And how many times do you have to get a flu vaccine? Every year. Why do you need to have a flu vaccine every year? Because there's changes in the virus itself. All right? Which will allow it to gain entry into cells, or it may allow it to transfer from one host to another, right? So that maybe it goes from pigs to humans or birds to humans. Okay? Those changes in the virus, because what ends up happening is virus infects a cell or host, it integrates some of the changes that occur. It may integrate parts of the cell of the host cell genome. It may integrate mutations may occur during translation or transcription that allow it to for emerging properties. All right. There's some debate of where viruses come from or how they originate, and then some candidates for where they come from in terms of plasmids, if they are integrating plasmid DNA, could be an origin for viral genome. All right, plasmids, transposons, and viruses are all what we call mobile genetic elements. All right, why are they mobile genetic? So genetic elements, they're pieces of genome, right, nucleic acid, that move from one bacteria to another, one host cell to another, or in the case of transposons, from one area of the genome to another area of the genome within humans. All right, so the ability to move and sort of move around the overall genome themselves. All right, and as you may have viruses are important pathogens, right, in animals and in plants. We've gone over several examples of what they cause. Measles, mumps, rubella, chickenpox, rabies, HIV, flu, the common cold are all viral pathogens. All right, diseases caused by virus. All right. Viral disease in animals. They make damage or kill cells, right, by causing release of hydrolytic enzymes. All right, some viruses cause cells to produce toxins, which then lead to your disease symptoms. All right, and that's sort of part of the influenza portion. All right, and then other viruses have molecular components, such as your envelope proteins, which actually are toxic to cells and to the host. All right, we know what a vaccine is. It's a way of conferring immunity against the virus. All right, so you take a harmless derivative of a pathogenic virus that stimulates the immune system. All right, and then you end up with an immune function, defenses against that pathogen. All right, so candidates for vaccines will be killed viruses, proteins of the envelope, all right, that will prevent of the envelope proteins, capsid proteins are all what we call immunogenic or antigenic. They cause an immune response in humans, and that's really what your vaccines are composed of. All right, viral infections cannot be treated by antibiotics because they don't stop the growth. All right, because viruses use your host cells. There are some antiviral drugs, all right, which prevent production of viral proteins, all right, but they can only help to treat, not cure, viral infections, right? Usually help to treat and prevent the spread and reproduction of viruses, all right? One of the antiviral drugs that for HIV, all right, targets the reverse transcriptase enzyme, right? So that it can't be produced from one, it helps prevent infection from one cell to another, right, based upon action, all right? HIV, emerging viruses, basically is just a way of characterizing new viruses that are coming up. HIV is an example, all right? Before about 1982, we didn't know what HIV was, and then there were populations of individuals that were developing classic diseases and dying normally from pneumonia, classic symptoms, and then it came about that it was actually a viral infection that was being passed from individual to individual. All right, West Nile virus is a more recent example. It first appeared in North America in 1999, and now has spread to all 48 states and is passed by mosquito progression, mosquito transmission. All right. All right. In 2009, even more recently, you had an outbreak or an epidemic of a flu-like illness, which occurred in Mexico and the U.S. Right. We call that H1N1, also known as swine flu. Right. Spread rapidly, causing a pandemic. Right, or a global epidemic. And part of this rapid nowadays are in current times with ease of travel from sort of global travel, airlines, we can pass global pandemics, right? Or pandemics are even more prevalent and easier based upon sort of travel between countries. All right, and sort of screening that occurs. Right, there's your H1N1 influenza virus. H1N1 is a characteristic based upon their glycoproteins that are found in the envelope. All right, you'll see that in a second. And then they had screenings so that there wasn't. All right, and then you end up with three processes contribute to the emergence of viral diseases. Mutations of existing viruses, which is why you get an influenza shot every year. Right. And then this is especially high in RNA viruses, all right? The dissemination of a viral disease from a small isolated human population allowing the disease to go unnoticed before it begins to spread. All right, so you start in one area, it's isolated, and then allows spreading. All right, and then spread of existing viruses from animal populations. All right, about 3/4 of new human diseases originate this way. This is also what's called a zoonotic. All right, a zoonose or zoonotic disease is one that can cross species. All right, it basically goes from animals to humans. All right, and there's some debate and evidence that supports that HIV was transmitted from other non-human primates to humans at some point in time. All right, somehow changed. All right, so you have your strains of influenza, H1N1, and virus 2 viral surface proteins, hemagglutinin and neuraminidase, right? And there's numerous types of H1N1, H2N5, whatever it might be. And they're identified by numbers, basically showing you that there's lots of different strains of the influenza virus that can change, and that's why every year you need a flu shot. Right? Viruses also infect plants. Important for plants is that this damages crops. All right? So, same basic structure, mode of replication as animal viruses, right? And most plant viruses are RNA genomes, and they have like a helical capsid, right? But there's over 2,000 types of viral diseases of plants that are known. It's important for industry and agriculture, all right? And then plant viral disease is spread by two major routes: infection from an external source. So you have here's a happy plant, right? It's infected by virus. Here's another happy plant, okay? And transmission goes from one plant to another. That's horizontal transmission, okay? And then you have vertical transmission, which now this plant that's infected produces seeds, which then leads to another plant that is infected with virus. That's vertical transmission. All right? So you have some that are transmitted from individuals from one to another. That's horizontal, or some that are inherited from progeny. That is vertical transmission. Okay, so we can talk about vertical transmission in humans and animals as well. All right, you can have a mother that is infected with HIV, can pass HIV onto her developing fetus. All right, that is a vertical transmission. All right, but from individual to an individual is horizontal transmission. Okay, questions on viruses. On Friday, you have a quiz on viruses. We're going to go over a little bit, another little bit of infectious agents on prions on Friday, and then we'll start the next part of the chapter of the book.
So the quiz on Friday, those extra slides that you get for.
I will post them right after this. Yes. And especially for your exam. Other questions? Okay, I'll see you on Friday.